Welcome to Wonderslate. Subscribe to our Telegram channel given in the description to updates and study materials. Topic for today's quiz is McCues on genetic code. Question number 1. Which of the following is not a feature of the genetic code? Option A. Triplet. Option B. Degenerate. Option C. Non-overlapping. Option D. Ambiguous. The correct answer is option D. Ambiguous. The genetic code is non-ambiguous. This means that there is no ambiguity about a particular codon. A particular code will always code for the same amino acid wherever it is found. Question number 2. According to pairing concept of wobble hypothesis, base I in the anticodon does not pair with option A, option B, U, option C, G, option D, C. The correct answer is option C, G. The wobble hypothesis does not permit any single tRNA molecule to recognize 4 different codons. 3 codons, A, U and C, can be recognized only when inosine occupies the first 5 position of the anticodon. Question number 3. The codon is _. Option A. Singlet. Option B. Duplet. Option C. Triplet. Option D. Quadruplet. The correct answer is option C. Triplet. The codon is a triplet. Singlet and doublet codes are not enough to code for 20 amino acids. Again, in case of a quadruplet codon, there will be 256 possible codons, which are highly excessive. Triplet codon thus is the minimum requisite, having 64 possible codons. Question number 4. Which of the following is not a chain termination codon? Option UA. Option B. UGA. Option C. UAG. Option D. UGG. The correct answer is option D. UGG. The universal chain termination codons are UA, UAG and UGA. UGG codes for tryptophan according to the universal genetic code. Question number 5. There is one amino acid for one genetic code. Option A. True. Option B. False. The correct answer is option B. False. In a triplet code for a particular amino acid, more than one word can be used. This phenomenon is described by saying that the code is degenerate. Question number 6. The first amino acid added by the tRNA is added to the anticodon _. Option A. AUG. Option B. UAC. Option C. ACG. Option D. UGC. The correct answer is option B. UAC. The first amino acid added is methionine, which is coded by the codon August. Thus, the anticodon for this codon in the tRNA is UAC. Question number 7. One base in an mRNA can be used for more than one codon. Option A. True. Option B. False. The correct answer is option B. False. One base in an mRNA cannot be used for more than one codon. This is because the codes are non-overlapping. Question number 8. The modification of which base gives rise to inosine? Option A. Adenine. Option B. Guanine. Option C. Uridine. Option D. Cytosine. The correct answer is option A. Adenine. Question number 9. The stop codon on the mRNA is read by _. Option A. Ribosome. Option B. tRNA. Option C. CRF1. Option D. mRNA. The correct answer is option C. CRF1. Chain termination codons WAG and AGA are read not by special tRNAs but by specific proteins known as release factors. RF1 and RF2 are found in bacteria and IRF1 in eukaryotes. Question number 10. The stop codon on the mRNA is read by underscore. Option A. Ribosome. Option B. tRNA. Option C. CRF1. Option D. mRNA. The correct answer is option C. CRF1. Chain termination codons WAG and AGA are read not by special tRNAs but by specific proteins known as release factors. RF1 and RF2 are found in bacteria and IRF1 in eukaryotes. Question number 11. In case of mitochondrial genetic code, AGA is _codon. Option A. Tryptophan. Option B. Arginine. Option C. Proline. Option D. Stop. The correct answer is option A. Tryptophan. In case of mitochondrial genetic code, AGA is a tryptophan codon, but AGA is a stop codon in the universal genetic code. Question number 12. With respect to the genetic code reading frame, which of the following is wrong? Option A. 5-3 direction reading frame. Option B. The code is non-overlapping. Option C. No gaps present. Option D. Flexible reading frame. The correct answer is option D. Flexible reading frame. The message from the mRNA is translated in a fixed reading frame, which is set by the initiation codon. Translation starts at an initiation codon located at the 5' end of protein encoding sequence. Because codons are non-overlapping and consists of the 3 consecutive nucleotides, a stretch of nucleotides could be translated in principle in any of the 3 reading frames. Question number 13. The wobble hypothesis was devised by underscore. Option A. Arthur Kornberg. Option B. Francis Crick. Option C. James Watson. Option D. William Asbury. The correct answer is option B. Francis Crick. In 1966, Francis Crick devised the wobble hypothesis to explain the observations regarding base pairing. It states that the base at the 5' end of the anticodon is not as spatially confined as the other 2, allowing it to form hydrogen bonds with any of the several bases located at the 3' end of a codon. Question number 14. The mitochondrial genome has _ stop codons. Option A. 1. Option B. 2. Option C. 3. Option D. 4. The correct answer is option D. 4. The mitochondrial genome has 4 stop codons. They are WA, WAG, AGA and AGG. But the universal code has 3 stop codons that are WA, WAG and AGA. Question number 15. The distribution of codon is made in such a way to minimize mutation effect. Option A. True. Option B. False. The correct answer is option A. True. Inspection of the distribution of codons in the genetic code suggests that the code evolved in such a way as to minimize the deleterious effects of mutations. For instance, mutations in the first position of a codon will often give a similar, if not same, amino acid. Question number 16. Fruitfly mitochondrial codes AGA and AGG codes for underscore. Option A. termination. Option B, tryptophan. Option C, serine. Option D, arginine. The correct answer is option C, serine. Fruitfly mitochondrial codes AGA and AGG codes for serine. In case of the universal codon AGA and AGG codes for arginine. Question number 17. Point mutation of the second place mutating A with T will result in a similar if not same amino acid. Option A, true. Option B, false. The correct answer is option B, false. Codons with pyrimidines in the second position specify mostly hydrophobic amino acids, whereas those with purines in the second position correspond mostly to the polar amino acids. Thus, when purine A is replaced by pyrimidine T, it results in an amino acid swap between hydrophobic to polar amino acids. Question number 18. With respect to the universal genetic code, pick the odd one out. Option A, UA, glutamine. Option B, UGG-tryptophan. Option C, ARGA, stop. Option D, CUG, serine. The correct answer is option B, UGG-tryptophan. UA, AGA and CUG are variations found in the universal codon, whereas UGG is not a variation. UA, glutamine codon in some unicellular protozoa, whereas codes for chain termination in the universal codon. AGA, stop codon in mammalian mitochondria but is the universal code for arginine. CUG, serine codon in the yeast, Candida, but is the universal code for leucine. Question number 19. Which of the following genetic code shows ambiguity? Option A, CGU. Option B, OG. Option C, GAC. Option D, UGA. The correct answer is option D, UGA. UGA is one of the universal termination codon, but UGA codes for tryptophan in mitochondrial genetic code. Question number 20. The mitochondrial mRNA code for methionine is _. Option UA. Option B, AGA. Option C, A. Option D, OG. The correct answer is option C, A. The universal mRNA code for methionine is August. The mitochondrial mRNA code for methionine is AWA. Thank you for watching the video. If you'd like practice this quiz, then download the Wonderslate app now. Link in the description below. If you have any suggestion for us, feel free to leave comments. If you liked the video and want to see more of it, hit the thumbs up and don't forget to click on subscribe button to get more updates of the future videos.